Hello, my name is Sarrazin-Gendron. I am at McGill University, and my talk is RNA 3D module identification in sequences with structural context sampling. So this is a bit of a specific take on RNA structure prediction, where we focus on specific substructures called RNA modules. Those modules are recurrent networks of non-canonical base pairs, so tertiary structure interactions, within loops, so unpaired regions of the secondary structure. Those loops are essential structural organizers and ligand binding center, and getting them right is essential in building a 3D structure from sequence that is reasonable, as was shown in different RNA puzzles contests. So there have been some efforts in identifying those 3D modules from sequence. Unfortunately, we cannot do so without using statistical methods because the energy models we use for secondary structures are not applicable to those tertiary structure interactions, which are a lot weaker. So we present the software base pairing 2, which is to this day the most efficient and a software that is at least as accurate as alternatives for identifying those 3D modules in sequences. And its workflow looks like follows. So the input is the RNA sequence or alignment. Then we sample the secondary structure. So usually we will take maybe 1,000 or 10,000 structures, which we will look through one by one. We will extract each loop of these secondary structures and try to match them to a database of modules through a size-based match. So basically, if we have the secondary structure, we have a three-way junction here. Well, we try to see if we have a three-way junction in the database that matches it. If we do find a match of a right size, then we will use a Bayesian network to score it. So the idea of this Bayesian network is to encode the dependence relationship between partners of a base pair. So if I have a canonical base pair and I know one of the nucleotides, I can infer the other nucleotides. So the idea here is just to encode those relationships for all types of interaction. And then this allows us to store all the sequence information of the module. So a module is, at the end of the day, a graph of nucleotides, where here the nucleotides are nodes and the edges are base pairs, and a sequence logo, which contains the statistics of the sequence at each position of the graph. And then this graph will match a local 3D structure. So that's what the Bayesian network allows us to score. And then our output score is based on this sequence match statistical score, as well as some measure of how common this secondary structure that allows the loop of the right size is in the ensemble of structures. So in terms of results, we tested on two main data sets, the RNA 3D Motif Atlas and RNA 3D Motif, which are two of the biggest methods to agglomerate modules. And we showed that there is a difference in distribution between hits on true module locations. So when we test a base pairing on a sequence that we know contains a module, and scores on false module locations when we test base pairing on a sequence that does not contain a module. So given these two distributions, we can establish a cutoff score that allows us to obtain a reasonable sensitivity and specificity, allowing us to reach F1 scores in the 0.93 range. The biggest challenge of this method revolves around having to deal with this size match, which is not optimal because in practice, a base can be bulged out and preserve the 3D structure. But nevertheless, for a large majority of the motifs we found in those databases, this assumption allowed us to represent one cluster of similar local 3D structures with a single graph. So it was not an assumption that costed us too much. So if you have any question, feel free to reach out to me by email. Thank you for your attention.